Okay, it's 4 o'clock, and I think we're gonna start sharp 4 o'clock. First of all, I have to do like this. Okay, this is the program. D'abord, bienvenue tout le monde, et certainement la famille, parce que c'est Alain, je connais Alain un tout petit peu, mais pas trop bien. So welcome to everybody. I think it's good. First of all, two things. We're still under the COVID-19 guidelines, so we asked what we have to do and not have to do. So unfortunately, we have to keep up the mask. Okay, so be it. Second, if you have a mobile phone, please switch it off. You're isolated from the outside world for about 2 hours. And afterwards, you can save the world, whatever you want to do. A very hard welcome. I'm very pleased that we have Alain for the Collens Francqui chair, they call it. And I'm going to shut up and give, with a lot of pleasure, the word to Hans. Hans, up to you.
Okay. Thank you for the introduction.
Short introduction, yes. First of all, I would like to call you welcome here during, you don't hear me? Better, this one? Okay. So I would like to welcome you on this special occasion. This is that's not Francqui, it's now the Collens Francqui. It's you know, they put a new name on top of it. And this is given to a person that is, you know, kind of an excellent one at level of research, but also education. And normally it's very difficult for us to find a person, you know, that could fit. But when last time when they were asking at the faculty, I darkly had a kind of a person in mind, so I didn't have a lot of problems to, you know, put my thoughts on Alain and just you know, ask if he would be a good candidate. And I think the whole faculty agreed with that. So that's why this medal will go to Alain this time. Before, of course, we go to the inaugural lecture from Alain, I would like to give a little bit background. Who is this person? He looks gentle, but we want to have a kind of a background knowledge on the person himself. So I was surprised about the age. 67, I'm from 63, so we're not that big difference. So he was born in 67. He got his DVM, so Doctor in Vet Medicine degree in Liège. As you will see, Liège is the place where he got all his talents. So he got in 91 his degree of Doctor of Vet Medicine. In 95, he got his PhD there, and that was on a topic in vitro study of the interaction between bovine herpes virus 4 and bovine host cells. So you can see already bovine herpes virus 4, so it's a mammal down there. Then he went to, and this is on the promotership of Paul-Pierre Pastoret, a very important person for Alain, I know that. Then he got in 98 a special title, which we don't have at the Flemish in the old days, yes, but not now anymore. So the Agrégation de l'enseignement supérieur en sciences. So this is something special for the balloon part. And this was, you know, he did a study, and you can see there comparison of intracellular and extracellular particles of vaccinia virus. And there's one big expert on that, and that is Smith in London. And that's where he got training there. And I think he did quite a good job there. And it was a kind, I call it a hybridization at that place. Then in 2009, and then all the work started to come because he became a full professor at Laboratory of Vaccinology and Immunology at our sister faculty in Liège. And as you can see, he got several awards in 2009. Prix Centre d'études Princesse Josephine Charlotte, study of host herpes virus interactions, still at that moment mainly working on mammal herpes viruses. 2016 GSK Vaccines Award, the infection of carbacyprin herpes virus 3, homologous host virus model to study vaccinology and fundamental immunology. As you can see there, he got a prize, but this is already with some fish in it, because that's the love of his life at this moment. And in 2019, he got for the first time a chair Francqui at the University of Catholic in the Louvain. So nice list of awards. I know Alain already for a long time, and I was always, you know, very much intrigued by the way he worked. and especially his broad interest in a lot of things. So I gave you some list, understanding virus evolution, and this is for vets, this is interesting because you always have different species, different viruses, and you can compare those things, which is sometimes, if you compare with humans, there is boring, you have one. So it's, it brings always very nice new questions, and I like to think about it. So he's doing similar things. Pathogenesis work, we've seen vaccinology, immunology, but I think he's a real virologist if you look to his work. Okay. Unraveling the mechanism developed by pathogens to evade the immune response. So this is also very interesting. This is going back to the evolution, especially the co-evolution, the evolution of your virus, but also the evolution of the host. And if you look to the interaction of your virus with the host, then you can, the virus will learn you a lot. And one of your lectures will, that will be the topic of one of your next lectures. So I'm looking forward to that one. And the last thing is identifying and exploiting innate adaptive immune responses, able to confer protection. And there, this is the point where his vaccine development comes in place, which is, I think, extremely important. If people now think always with the coronavirus, COVID-19, it's so easy to make a vaccine. For coronaviruses, it is, but not for all viruses. So sometimes you really need good brains to try to find out which branches of the immunity that you should stimulate to come with a strong protection. So this is part of his work. He did a lot of more things, but so his output, his co-promoter of 20 PhD students. I know what it is to train young people, and I can tell you there are very strong persons in between them, a few professors, so he can be really proud on what he developed. And then also 163 publications, and they're always top. So veterinary virological, so he doesn't like to stay in the Q4. He's always getting in the Q1. So congratulations to that, Alain. We will not go over all his list, but just to see, sometimes in the veterinary world, they always think of NAS paper, but for Alain was not an issue. So you see here a few NAS papers, Journal of Virology. And if you look into the topics, so he was here, I call it, you cannot see it behind his bar, but this is his life outside the water, I call it. So this was all concerning animals that were living, so viruses that were living, that were replicating in mammals, like, as you can see, African buffalo, then here you have the cattle. This was vaccinia virus, also mammal. And this was something special, a qualifying herpes virus, which is normally, going into wildebeest, the gnus. And then this was showing that he was looking for a change in his life, going away from viruses in mammals. Suddenly he found some things in ticks. And then I was thinking, I was looking to his paper. I said, what the hell is he doing now with ticks? So I was thinking he would continue in ticks and we would lose him into the tick world, in the insect world, but into the tick world. But he, came back, and that's then his inside the water period. I really like his work. And what is very pleasant to see is that he's really doing that with a big joy. He's not looking to opportunistic things, because if you're an opportunist, you don't go into this thing because there's not a lot of money to find. So he really, he enjoys his life. He just grabs this thing, and you can see on these top papers, he's making progress and making already vaccines and things like that. So I'm really proud that that there are still some veterinary virologists behaving like veterinary virologists and doing a lot of progress in that. And this is, this is what you get if you get an email from Alain. This is what you have at the bottom. A lot of people are just putting a list of their, of their publications or whatever, and how important they are. This is what you get from Alain. Let's enjoy science, and this is how he behaves. He's just always, he's always full enthusiasm to talk about science. Out of the box thinking. So if you want to have a kind of a brainstorm session, and you want to go into the depth, you can always talk to Alain. Use of frontline technologies. Veterinarians normally are very scared of technologies. We're always, trying, to avoid them, keep it simple. But Alain is one of them. Bioluminescence, whatever he thinks, he's doing it. Never give up. I think that's a very, I wanted to write down pit bull attitude, but I think that was a little bit too strong. But I recognize, and I see it also with the students, he never gives up. And then as a student, it's very hard, tough, of course, to cope with the boss like that always says, come on, because to deal with this person is extremely difficult. Interested in all scientific fields, whatever human, whatever animal you have, you can always talk with him. Strong promoter. I told you already, he has very, strong, even a few professors coming out of his hands. And very important, and I put that as last but not least, he's a veterinary scientist, and he's really proud on that. And I appreciate that, because we know there's a lot of, we just discussed that, there's a lot of pressure on the young people, especially at the level of trying to get money and try to gather some money and try to have projects accepted. A lot of people are following the human way because they find there more money, or that's what they think in the beginning, of course, but it's extremely tough to keep on going in the research as a virologist. But I see he's coping well, and with this I will stop it. It's a little bit longer than the Dean, my presentation, but I wanted to put you in a good position so that you can give your presentation. So Alain today will give an inaugural lecture, viruses, the good, the bad, and the ugly. Alain, the floor is yours. I will give you the microphone.
Yeah. Training after I can work at the quick.
No, like Madonna now. Yeah.
McDonald's.
Here is your pointer.
Yeah. Okay, good. Okay. So far, so good. Can everybody hear me? Yeah, that's a first good start. Dear Prorector, dear Dean, professors, students, ladies and gentlemen from the general public, dear friends, dear member of my family. It's a very great pleasure to be here today. And first of all, I would like to thank the faculty of veterinary medicine from the University of Ghent and the Francqui Foundation for selecting me as the laureate of this prestigious award. I'm interpreting this award as a recognition of the work that our lab has been doing over the last few decades, but also as an encouragement to pursue the type of research we are currently doing. Every single person needs positive feedback from time to time. There is a long list of reasons why I'm particularly happy and proud to have this prize in this faculty, and I'd like to stress two of them. The first one is quite obvious. This faculty is ranked #1 in the world for many consecutive years. Yeah, you can applaud yourself. And this makes this award even more prestigious. And the second reason is for the man who proposed my name. So, you know this guy. So as Hans mentioned, he's just a little bit older than me. So I had the chance to observe his career throughout my life. And throughout these years, I've learned to admire this guy because he's a guy who do great science, numerous studies, high-level studies. He always fight for science, and this is becoming increasingly rare. And he has always been expressing a high level of passion for science. And this altogether means a lot when I knew that he proposed my name. Hans and I, we have a few very good memories, and one of them is certainly that special concert that our respective university organized. And I don't know who select Hans and I to talk to our king, but this was a marvelous evening, especially at the end, and it ended very late, and I should say very early. Being laureate of the Francqui Chair is a very, very special scientific award in the sense that the laureate has to give lectures. And these lectures should form a kind of journey into a scientific topic. So as far as I'm concerned, I decided to talk about viruses as model of biology. And I will address a different topic through the three ordinary lectures. So in the first lecture, I will talk about the origin and the evolution of viruses. This is quite classic. But I will also talk about the role of virus in our own evolution. Very few people know that some of our genes are derived from viruses. So part of you is virus. In the second lecture, I will address what has been fascinating me for more than 30 years now, is the virus-host interaction. This is a fascinating field. And as soon as you start to study these interactions, you very quickly realize that these viruses have been studying biology far longer than us. And you can, as Hans mentioned, learn completely unknown aspect of host physiology by looking at viruses. And in the third lecture, I will try to give you a touch about applied research. When you grow up in a university, you are educated based on fundamental research. But when you become older, like I do, you want to develop solutions for problems. And this leads you to applied sciences. And I will, in the last lecture, describe biotechnological innovation that have been developed based on viruses. So these are for the ordinary lecture. Now, a Francqui share also implies an inaugural lecture, a special lecture, and that's the one I'm going to give you today. So I received some recommendations for this lecture. And basically, I should try to reach 3 goals. The first is to tell you an important or several important events in my life that switched my interest for science. Not easy. Second, I need to stimulate the young generation, their interest for science, and try to give you good advice to be successful in life. I certainly care about your future because you don't realize, but you will have a huge privilege in life. You will contribute to Hans and I retirement. And for sure, you are the generation and yours as well. So we really want you to be the most productive and bright generation of human history. And our pension rely on you. That's very important. And then the third goal I should try to reach in this lecture is to stimulate critical thinking. So let's try to address first the 2 first items. An event in my life that influenced my interest for science. Okay, so for most scientists, it's difficult to identify a specific moment that switch on your interest for science. In my case, I remember very well, this happened when I was 16 years old. So 16 years old, you won't believe me, but I had long hair, and I bring a picture. That's my identity card when I was 16 years old. I just came out of the hairdresser not to look too much like a girl. And at that age, I was not interested at all by my studies. And I was actually suffering from this education system that was, in my point of view, completely dogmatic. I had in front of me people, my teachers, claiming that they knew everything, and they were asking me to remember a question and an answer, and to swallow this answer and to vomit it at the right moment. So I was really hating the education system, and I was doing just the minimum just to make sure that this pain will not last too much and 1 more year. But nevertheless, at that age, I had a very, very strong motivation to go every day at school. And my goal was to fancy the most beautiful and interesting lady I've ever met in my life. So every morning I was going to school with the hope to become the boyfriend of this young lady. And so I was looking for opportunities to approach her and to fancy her. And one day, by chance, I realized on the board that she registered to attend a seminar in Brussels, a seminar organized in the context of our course of biology. So to get the point, I registered just to have an opportunity to approach her. And this is how I went to the conference given by this great man, Professor Christian de Duve. You most probably know him. He got the Nobel Prize for his work on mitochondria. So this guy came on stage, extremely charismatic person. He said a few words and the room was completely silent. And he presented himself and said, I'm Christian de Duve, I'm a scientist, and I spend my day addressing questions that do not have answer yet. I spend my day addressing questions that do not have answer yet. Can you believe it? I was living in a world with teachers that they claimed they knew everything, and then a Nobel Prize, a guy defined like a smart guy, was coming and said, there are plenty of things that we do not know, and I'm living there. I'm living at the border between the known and the unknown, and I spend my day trying to make the unknown going backwards. This was a total revolution in my head. So the next day I came to school And I asked to talk to the professor of biology. And I told her and say, hey, Madame Vioff, this is her name, she's dead, so I can pronounce it. And I say, do you know something happened yesterday evening? I think I want to be a scientist. And this lady, she was good, nice, but she looked at me and she smiled. She didn't laugh. And then she explained, she said, but Alan, you need to go to university to be a scientist. She didn't continue her sentence, but she was clearly meaning, you stupid, you will not reach there. And about a year later, when they asked my mother, you know, at that time we had in the Walloon region called Conseiller d'Orientation Pédagogique. This is a group of terrorists. And my mother went there to know what I could do in life. And then she explained that I wanted to be a vet or a scientist. And the lady laughed and said, now, come on, be serious. He's strong. You have a farm. Let's convince him to take the farm. But this guy cannot go to university. And I want you to remember two advice from this story. And this is for the young generation. One, when you have to take decision for yourself, never allow someone to take decision without your consent, and take the decision yourself. You are the master of your life. You are the person who has to decide for your future. Don't let somebody else decide it for you. That's one. And the second, it's more important, you have to keep in your life some room for serendipity. We try to live a life which is rational and it makes sense, but sometime you will realize that very important moment in your life will occur at the moment that you don't expect them. Just like me, when I went to listen to Christian de Duve, I was not going there to listen about mitochondria. I was going there to fancy a beautiful blonde. And it came out to be a major moment of my life that switched on as an intellectual interest that none of my teacher managed to switch on before. So remember this. And there's a beautiful quote from Steve Jobs that illustrates this concept. And in this quote, he used the word dot to mention an important moment in life. And he said, you can't connect the dots by looking forwards. You can only connect them by looking backwards. And you have to trust that somehow your dots will connect in your future. That's very important. So concerning my list, my to-do list, I've already told you one event in my life that pushed me to science, and I give you free advice. I will give you more later for free. So now let's move to the masterpiece, the inaugural lecture, and a topic that should stimulate critical thinking. So I thought a lot about this, and I had the idea to provoke you with a question. My question is, do you really believe that all viruses are bad biological entities? Of course, you will reply, yes, of course. Where have you been over the last 2 years? The entire planet has been suffering with this virus. So of course, viruses are bad things. And my goal today is to demonstrate that this is wrong. And that most frequently in biology, when you ask a question, there is no a black and white answer. So this title sounds like a movie title. So it's viruses, the good, the bad, and the ugly. Remember this Western with these three guys, there was the good, the bad, and the ugly one. And this brings me to the reason why you receive a ticket when you came in. Everybody has a ticket? Who doesn't? No, you have one? So this is a champagne competition. So you'll write your name on it, and then I will, in my talk, mention 5 different movies, or more than 5. And you have to detect them and write them down. And at the end, we will select the winners. There are 7 winners. The first one will have a very large bottle of champagne. The 6 others will have a normal bottle, but all the bottles are good. You will not receive them today because, as usually, when you don't do everything yourself, someone forget to do what you ask for. So, but Hans will take care of the bottles. I will bring them next Friday, or if you come next Friday, you can pick up your win if you are the winner. Okay, so let's go. The good, the bad, and the ugly. So that's the first title. You can write it down. And what I will try to convince you in the next hour is that when facing a question, if are viruses all bad or good, you don't have a black and white answer. And in fact, there are more than 50 shades of gray. You got it? In between these two extremes. To convince you about this more than 50 shades of gray, I decided to tell you 5 stories of virology that I like very much. And through these stories, I will be able to convince you that there are good viruses and bad viruses. And my first story is entitled The Ugly Virus, the cow, the good virus, and the scientist. So let's first introduce the first character of my story, the ugly virus. So the ugly virus is variola major. It's also called smallpox. That's a human killer. This virus has killed humans for at least 3,000 years. We have evidences that it was already killing people during the Roman Empire and probably at least 1,000 years before. This virus, I'm not talking about prehistory. This virus was eradicated in 1980. So people like of my age were vaccinated against smallpox. When you heard on TV that COVID is the worst viral infection that human has to face in history and blah, 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 that's simply false. COVID killed only few percent. This guy was killing 50 to 80% of affected people. So this was a beautiful way to regulate human population before contraception. And this virus, and this is very important, please be careful, otherwise you will not follow the rest of the story. This virus, smallpox, belonged to a genus. In virology, a genus is a kind of family. These regroup viruses that derived phylogenetically from one common ancestor. So if we have the possibility to go back in time with a time, don't worry, it's okay, I'm still alive. Yeah. If we go back in time long enough, probably millions of years, we will be on Earth with a single ancestral virus. And throughout evolution, this ancestral virus has speciated, has formed different viral species that adapted themselves to different animal species. And this is why how you reach our days with one member of this genus that is adapted to cow. This is cowpox, vaccinia as well, monkeypox to monkey, and variola major was adapted to human. But very important, throughout evolution, these different viruses conserved one of several molecules from the ancestor unchanged. So that means that all these viruses have in common few proteins that are pretty conserved, with the consequence that if you are immunized against one of these viruses, you are in fact protected against all of them. And this is very important and different from what you observe with other viruses, where within a viral species, you have antigenic differences that do not confer protection. But here is clearly the opposite. These viruses, even if they form different viral species, because they have some molecule identical between them, they confer a cross protection between them. So let's continue with some description about smallpox. So if you don't like horrible picture, close your eyes. I will tell you when you can open it. So I will show you a picture of a lady who was affected by smallpox. You see her here. She fortunately survived. So this virus is a terrible virus. It's very resistant in the extracellular medium. If you put smallpox or even vaccinia on a dry surface, it will remain infectious for weeks. And you can contaminate yourself in 2 or 3 weeks by touching this material. So again, not like COVID, which is a quite unstable virus. So this lady probably contaminated herself through the oral route and the respiratory route. And the virus, when it entered the body, replicate fast and furious. Got it? It replicates into the body and then localize on the skin and the mucosa. And on the surface of the body, it will form pustules where the virus replicate. And each pustule that you see here will contain about 1 million or 100 million of infectious particles. So with 1 or 2 of these pustules, you can contaminate the entire world. Now, this lady survived. And when you survived from smallpox, of course, you were expressing the fact that you were infected on your surface. Nobody asked this lady one day, hey, by the way, did you get infected one day by smallpox? It was obvious. She was covered by scars. So very quickly, thanks to these scars on the surface of the body, people realized that once you have been infected by smallpox and did you survive, you were protected for life. A correlation was established about the fact of being infected in the past and being protected for life. The concept of we called immunity. And the fact that people noticed that you were protected for life after the first contact, and that they were convinced that one day or another they will be exposed to variola, led to an incredible procedure. People thought, well, one day or another I will be facing smallpox. So let's know as soon as possible if our kids will survive or not. Imagine you are the parents of a boy, you want to offer him expensive studies in Ghent to become a vet. You want to know before you go to the study and you invest the money if he will survive when he meets smallpox, right? So what people who did, they were deliberately inoculated the virus to people. And this was called variolization. So doctors were doing variolization. So when you had a boy or a girl, you wait that he is in good shape. And then at one stage, people were calling the doctor and say, okay, let's try the variolization. And we were immunizing the boy, and there was two outcomes. Didn't survive, he died, that's it. Or he survived, he will make smallpox, will survive, and then he will be protected for life. So this process was initially invented by the Chinese and was imported in Europe. And I'm now bringing you at the end of the 18th century to introduce the second character of my story, the scientist. This is Dr. Jenner, he's an MD, working at Berkeley in the Gloucestershire. So it's over here. It's a beautiful region of England. And it's important to mention that he was working there because Jenner was treating local people that were mainly farmers producing milk. And on the other hand, he was treating rich patients that were coming there to take some rest in the beautiful resources of this area. And progressively, Jenner came to a conclusion. He was convinced and observed frequently that rich people were more affected by smallpox. They had more scars on their face, while people from the farm, they were pretty okay. And this is a very important step in the mind of a scientist, that you don't know why, but you start to be upset by something that you observe. And this is a kind of phase where you imprint your brain. Then your brain is waiting for an event that will give you the idea. And this event can come everywhere. When you're swimming, you go to cinema, whatever. And one day, this event arrived for Jenner when he came into the farm of Sarah Nelms. And this is the lady that you see always on the painting that represents Jenner. And so he came into that farm, shake the hand of Sarah Nelms, and he realized that she had lesion on her hand. Lesion that looks like smallpox. So he immediately thought, well, Sarah, you are developing smallpox. Then make a physical examination of that young lady and realize that she didn't have lesion elsewhere on her body. So this was very intriguing. Lesions that looks like smallpox, but only localized on the hand. And so he discussed with the girl, and the girl informed Jenner and replied a very important sentence. He said, this is the lesion I got when I milk my cow and when they have similar lesions. So you have to realize that we are here at the end of the 18th century, 1 century before Pasteur discovered microorganisms. So Jenner didn't know that he was working with viruses. He ignored completely the existence of bacteria, etc. So you have to put in perspective how smart this guy was to be able to build an hypothesis without knowing the existence of microorganisms. So nevertheless, he set up an hypothesis and saw, okay, the cow has some lesion on her udder. Ladies are milking cows, and probably they contaminate themselves from these lesions. These agent, didn't talk about agent, but this agent could induce a benign localized infection in cow and do the same into human. But then his hypothesis was that the exposure of human being to this material coming from cow and that caused a benign infection could confer a protection against variola, smallpox. So kind of immunization, what we call nowadays heterologous vaccine. You take one agent and you confer protection against another agent thanks to the antigens that are coming. Now in science, where there are many, many moments which are very exciting, and certainly the moment you have an idea, that you have an hypothesis, is a very, very exciting moment. But soon after, you have to work and to develop what we call a model, an experiment to test your working hypothesis. And so this is what Jenner did. And just to remind you again, so variola major is here, and the virus, Jenner didn't know that, but the virus that were infecting cow are cowpox or vaccinia. We don't know which one he got, but these two induce localized lesions in cow and do the same in human when they are transmitted. But because these two viruses belong to the same genus as variola major, they confer cross protection. So hypothesis, then experiment. How do you make an experiment? Well, it's very simple. You need to have material from the cow. So we went to Sarah Nell's farm, identified the cow which contaminated Sarah Nell's. This cow is called Blossom. She became famous. Collect the pus, the exudate from the lesion. Then, using a needle, inoculate it to a young boy. You need a seronegative young boy. How do you identify a seronegative boy at the end of the 18th century? Well, it's very simple. You put it naked and then you observe the skin. There is no lesions, so it's a seronegative boy. So inoculate the boy with the material collected from the cow, wait that the virus from the cow replicate locally, and then we challenge the boy with lethal material collected from a person dying from smallpox. So this sounds like an horrible experiment. This is a criminal. He took infectious material from a person dying from smallpox and he deliberately inoculated to a young boy. This is unbelievable. Always put things in the right context and historical context. What Jenner did here is just varialization. He was doing this every day as a normal medical practice. So there is nothing really, really special. So you repeat this experiment and indeed demonstrate that if you inoculate this material coming from cow that induce a local and benign lesion in human, this confer later a protection against the lethal smallpox virus. And he published this in 1998. Now I want to mention something interesting about this publication. One day I was at the Royal Academy of Medicine in Brussels, and I met a very important guy working in a very important company doing human vaccines. And we were discussing about the discovery of vaccination. And this guy was arguing that Jenner knew that he was working with virus, which is wrong for sure. Jenner is 1 century before Pasteur. No way he knew the existence of viruses. But the guy was insisting and say, yeah, come on. In this publication, he's using the word virus, which is not possible. So the next day, it was a Sunday morning, I tried to find the publication on the web, and I found it. And surprisingly, I read cowpox virus. Can you believe it? This is the name that the ICTV gave to this virus a few years ago. How could Jenner use the term virus 1 century before the discovery of microbiology? This is completely mad. Of course, there is an explanation. The word virus, Latin word, at the end of the 18th century, was not used to describe a biological entity that replicates in 2 cells. It was used to describe exudate, pus. So when Jenner was saying cowpox virus, he was not meaning the virus cowpox. He was talking about the pus, the exudate that you found when the cow had cowpox. And this is quite funny and interesting. But as a scientist, you have always to be sure that your judgment, your interpretation is not biased. And you will do, and I do everyday mistakes, because we are not able to get free of our intention, the influence we have. So this is very, very important. Remember this story. Now, you heard in the press that there are anti-vax for COVID. It sounds strange. This has always been existing. This is a cartoon of the beginning of the 19th century against vaccination discovered by Jenner. You see, people were claiming that if you administer material from cow to ladies, for example, they will start to have horns on their forehead. This can happen sometime without being vaccinated. And so that's always been like this. Now, in my email signature, sometime I had a sentence, I always said, let's enjoy science, but I always say also, the good always win. That's true. Sometimes it takes millions of years if you look at evolution, but the good always win, always. And so the vaccination discovered by Jenner prevailed and replaced progressively viralization. Now, you imagine the time of Jenner, the source of vaccine consisted to go in farms and search for cow that had lesions on their udder. It's of course a very limiting source of vaccine. So people very quickly try to find an alternative to this. And they had the idea to use the material growing into an arm of a vaccinated person to vaccinate others. So what they call the arm-to-arm passage, which was quite good because then you vaccinate someone and you ask this person to come in 2 weeks. She has big pustules and you use the exudate from that person to vaccinate other person. So that was good. But of course, it's potentially a source of problem. If the person that you use as a donor of the vaccine has tuberculosis or syphilis or whatever, you contaminate the vaccinee with the contaminating agent. So this was not perfect. And so came the vet into action. And history tends to forget and to neglect the importance of veterinarian into human medicine. And I will show you a very important role played by the vet. The vet thought, okay, the arm-to-arm is too dangerous. There are very high risk of transmission of disease. Why not using the cow as a bioreactor? And so for more than 1.5 centuries, the vaccine has been produced as followed by vet. The cow were put down, then they were shaving the abdomen of the cow, and then using this horrible tool, cannot see it, but it's a stem composed by needle. They were contaminating the tip of the needle and inoculating all the skin of the cow. Then the cow was unwrapped in clean tissue. You put back in the farm. You clean the cow every day. And 2 weeks later, you put the cow down again, and all the site of inoculation form lines of exudate. Then you just need to collect the pus on the surface of the skin. The vaccine for human has been produced like this for 1.5 centuries. Amazing. By vet. The picture I showed you a minute ago is actually a picture, and you can read it in French. It said, Monsieur de Givre, directeur de l'école vétérinaire de l'État à Bruxelles, Kuringen. So this vaccine was produced there. So this is how the vet solved the problem. Of course, we still have some biosecurity issue because sometimes the cow has brucellosis or tuberculosis, but it was less problematic or less frequent than with the human-to-human passage. After, of course, the cell line appeared, QC of material, and people were able to produce clean vaccine. And of course, the lesion that you find on the arm of your parents or people of my age, that's the arm of my father, you see that he has been vaccinated twice in his life against smallpox. And this vaccination apply on all human beings, and it's very important. You can eradicate a pathogen only if everybody play the game. If everybody is vaccinated, then you eradicate the disease. And this is what happened with this virus, this killer virus in 1980. And on the 8th of May, 1980, the World Organization declared this virus eradicated from the planet. So the virus is now just existing in 2 laboratories in Atlanta and Novosibir, but does no longer exist in the wild. And that's the reason that people who have a birthday after 1980 are no longer vaccinated against smallpox. So that's for my first story. And you see that just with this first story, you have a bad virus, the ugly virus, but you have also a good virus that help us to get rid of the bad one. Now, in the second story, I will reuse one of the good virus. So the vaccinia virus here, which is one of the virus from cow that has been used to vaccinate, I will recycle it in the second story. And the second story is entitled The Two Viruses, Master Fox, and the Three Scientists. Beautiful picture. Can I move the, yeah, that's better. Oops. Okay. So you know already one of the two viruses, it's vaccinia, the virus that was used to vaccinate against smallpox. Let's now introduce the second virus of this second story. The second virus is rabies. You know, this rhabdovirus, it has a bullet shape and is covered by a trimer of glycoprotein G. So this virus causes encephalitis that systematically kill the infected subject. In last century, end of last century, still in the 70s, we still had rabies in our forest. And this was rabies affecting red foxes. It's kind of paralytic form, killing very, very efficiently foxes. And it actually acts as a negative regulator of fox population, which is a good thing. Now, the problem was that these foxes, sometimes they transmitted the disease to domestic animals, or cow, or dogs, or cats. And these domestic animals could transmit it to humans. So this was a real health issue. People wanted to get rid of the rabies into our forests. On this map here, each little red dots represent a confirmed case of rabies. So at that time, it was a serious issue. It was quite frequent. So people decide to set up a vaccine strategy to eradicate rabies from our foxes. So how do you vaccinate foxes against rabies? If you ask some people, they will tell you, well, it's very, very easy. We know since Pasteur how to protect against rabies. You just need to make a couple of injections. So let's put advertisement in the wood, free vaccination against rabies, Sunday morning, chicken wings and barbecue sauce after vaccination. No foxes will come, of course. Then all the people say, well, let's catch the fox. Let's go in the forest and trap them and catch the foxes. If you think that you can catch foxes, I advise you to watch this movie, The Fox and the Ant. The strategy described for the fox in this movie is about 10% of what they are able to do. They're extremely smart. No one can catch foxes, all the foxes in the forest. It's impossible. They're by far too smart. So how can we do it? To vaccinate foxes without catching them? Well, you need to be very smart. And some scientists are very smart. And they decide to design and develop a vaccine that will work through the oral route. A vaccine that will put in a bait that you will distribute in the forest, and then the fox will find them and ingest the bait and vaccinate themselves. And this brings me to present the three scientists. And in my life, I had the chance to have two of them as boss. The first one is Jeffrey Smith. So Jeffrey Smith was my boss in Oxford. I never worked in London. He's a very, very smart guy. And when he was at Bethesda, he had the idea to genetically modify the virus. So he had the idea to take vaccinia virus, the virus that we use to vaccinate against smallpox, and to insert into the genome of this virus a gene coding for a foreign protein, with the idea that when this virus will be inoculated to a human being or an animal, it will vaccinate the person against smallpox, but we don't care anymore because the virus is eradicated, but it will also protect the person against the pathogen from which we inserted the gene. So this was very, very smart. The second scientist is Jean-Pierre Lecoq. I never met him. He is a molecular biologist from a Belgian molecular biologist working in Strasbourg. And he had the idea to take the gene encoding for the glycoprotein G of rabies and inserted it into the vaccinia virus. So you obtain a recombinant, a GMO, a genetically modified virus, vaccinia virus, that will when it grows into cells, will express the glycoprotein G of rabies. So when you inoculate this recombinant to an animal, the virus infects the animal and express the antigen from the rabies. So thereby immunizing this animal against rabies. So he constructs this recombinant. And then the third scientist is Professor Paul-Pierre Pasteuret. So he was my mentor of my thesis, a very great scientist. And yeah. And Paul-Pierre, he made very simple but beautiful science. Like, for example, demonstrating that when you drop a virus suspension in the oral cavity of carnivores, you induce a limited replication of the recombinant virus, which is sufficient to induce immunization. And he did this on badger and on foxes. And then he had to design a bait, a vaccine. And this is the most original form of vaccine. So the vaccine consists of a little plastic bag in which you have a suspension of recombinant virus. So this contains infectious particle of vaccinia virus encoding glycoprotein G of rabies. And then this is surrounded by fat and some rotted meat that will be extremely attractive for the foxes. And so for that, you have to write down, this is a regulatory vaccine, don't touch, just in case a stupid guy in the forest find it and decide to eat it. It's regulatory people, they sometimes very fun. And so what the vaccination consists to fly with helicopters above the forest and then to drop these baits over the forest. And you imagine what's happening below. The foxes are saying, what this Sinterklaas is coming. Yeah? We have free food today. Looks very, very interesting. Then, because they have an amazing smell, they smell and find the bait, eat it, disrupt the little plastic bag, inoculate the trout with this recombinant virus. The virus will replicate just few cycles. It's not harmful at all for the fox, but this will be sufficient to induce the expression of glycoprotein G. And by this way, the fox will be vaccinated against rabies. And the fox is lucky because he had a free snack. Now, do you remember this slide I show you? Each dot is a case of rabies. I will now show you the situation of after a single campaign of vaccination. Watch carefully. Boom. You see the effect? You don't need statistics. And the only dots that you see here concentrate around all borders and are most likely foxes that cross the border, because at that time, the other countries didn't want to apply this vaccination. After these results, everybody applied this vaccination and we get rid of rabies into Europe. That's the publication of Paul-Pierre in Nature. Now, let's think about this innovation. As citizen of the society and a scientist, it could be that sometime people will turn to you to have an opinion. And an opinion like, for example, on GMO. And the society tend to force us to have a kind of dogmatic positions, be against or be for GMO. And at the moment, yeah, everybody wants to be against. And they cite horrible example of GMO that it's okay to be against. But the problem is that when you are against GMO, you are against a vaccine like I developed, because according to the regulation, GMO means everything that has been genetically modified as the product of human being. So the vaccine that Paul-Pierre developed 20, 30, 40 years ago will not be accepted this time. And this is a problem. So as a scientist, you will have to stand up. And when people try to have black and white answer, don't give up. Science it's extremely rare that you can have a radical binary position in science. This is true for the virus, it's true for the GMO. Let's move to the third story. So the first story were on Earth, human and foxes. Let's go a little bit in the water. The third story is entitled Viruses as Doctors of Mother Nature. So you most probably know what this is. This is algae bloom. You have, if you like the green, I've red, and they're so impressive that you can see them from space using satellite. And what's the origin of this algae bloom? Well, you need to, first of all, some pollution, an important source of nitrogen and phosphorus into the water. Then if you have a calm sea, these molecules remain at the same place. And if the temperature is high enough, this will stimulate the proliferation of the algae, and you have an algae bloom. These algae can be harmful for the ecosystem. Most of the time they produce toxins, so it's terrible for the ecosystem. It's really an ecological disorder. Now, for centuries, people realized that these algae bloom, they always resolve spontaneously. And it always takes about 2 weeks. So, of course, if you have a big storm the next day, they are washed away offshore. But even if the sea remains completely calm, this phenomenon will disappear in about 2 weeks. And you will observe, if you watch carefully the water, that the algae will start to agglutinate and they will die. What's the phenomenon of this? Well, scientists look at these algae. This is an electron microscopy picture of an algae, a unicellular algae. So what I'm showing you here is an algae. And all the little pearls that you see popping out of these algae are viruses. This is a Ficotna viruses. It's a huge virus. They have about 1,000 genes. They have more genes than the smallest cells existing. So very complex virus. Now, if you think about the challenge for this virus is infecting one algae, then it has to go out of one algae, an infected algae, and bind and penetrate a non-infected algae. That's quite a challenge when you are in the ocean. So not surprisingly, through evolution, viruses that acquire the ability to bring together an infected algae and a non-infected algae will have a selective advantage. Well, this is what happened in evolution. These viruses acquired enzymes that allow them to express on the surface of the algae a virtual Velcro, a kind of glue, that when an infected algae moves into the sea and touch a non-infected algae, it's linked. And so the virus has just to make one step to move from the infected algae to the non-infected algae. And this explains why, according to time, you see this algae agglutinate and dying because the virus is inducing their agglutination thanks to the production of this glycan. I'll show you this. The picture is not too good. So this is a section through an algae. So this is the cell wall of the algae here. I'm showing with the pointer. Below you are in the cytosol of the algae. Above you are in the sea. This is the glycan that are expressed on a non-infected algae. So let's see how the cell is modified by the infection. Just 4 hours after, look at this. This is the reference, the control, mock infected, very short glycan. After 4 hours, you have this Velcro who has been growing on the surface of the infected algae that will act as a glue and make non-infected algae sticking to the infected algae. So thereby making the spread of the virus extremely easy. This has been demonstrated to be glycan because if you treat with glycosidase, you can remove them. Now, these viruses are really fascinating because if you look carefully where they perform the glycosylation into the algae, scientists realize that these glycosylation are performed outside of the ER and the Golgi. So they most probably represent a kind of ancestral form of biochemistry that occurred at the stage where Golgi and ER were not existing. So these viruses teach us really a lot about the physiology and the host. So second story, third story. Now I'm now moving to the fourth story, which is a very special one. And I call this viruses of insects as subject of fundamental and applied research. And what I will try to transmit in this story is the importance of serendipity in science and how from a simple experiment, from a simple observation, you can lead to completely unexpected application. Again, you can only join the dots by looking backwards. And these viruses infect insects. And I will talk about Spodoptera frugiperda. This is an insect pest that destroy legumes and different type of culture. It's a classical lepidopteran cycle. You have male and female, they mate, then the female drop their eggs on the leaf. Then you have a little caterpillar that came out of these eggs. And this caterpillar makes three things: they eat, and they eat. They spend their time eating. And they grow like mad. They multiply the weight by about 1 million in just a few weeks. Then when the caterpillar reach a critical stage, it will go down the plant, leave the leaves of the plant, go into the ground, and then transform into a pupa. Inside will occur the metamorphosis that you know, and turn the caterpillar into the butterfly. So this is a typical lepidopteran cycle. Scientists are, of course, studying insects. And one day a scientist realized that this colony of insect in his lab was dying, which is terrible because then you can no longer study insect. And he tried to understand what was the cause. And what he observed is that the caterpillar was virtually turning into liquid and was lysed on the leaf of the plant, dying from an obvious infection. So he called virologists and microbiologists. People look by EM, etc., and they found that these insects were infected by baculoviruses, an entire family of viruses that infect insects. And they also found that this virus is very, very smart, because it does not release the virus in completely free way. It actually unwrapped the virus in kind of occlusion bodies that will protect these variants from the light and from the dry. So you can, when the caterpillar die and virtually turn into liquid on the leaf is contaminated for weeks. So just waiting for a new small caterpillar that will eat the leaf, digest this inclusion body, and reinitiate the cycle. Another thing that people observe, and they say, well, this is weird. When my caterpillar are infected at the late stage, close to the stage where they should go down to the leaf, they don't longer behave normally. They remain on the plant. Just like if the viral infection was modifying their behavior. That's fascinating. Huh? There are people studying the behavior of caterpillar. Can you believe it? So these guys said, this is really weird. Then they went to scientists studying the behavior of caterpillar. And actually, there is one molecule that regulates the physiology of caterpillar, and this is the IgD steroid. This is this molecule. And so these scientists look at the steroid of infected caterpillar, and they realize that the molecule has changed. So in non-infected caterpillar, it looks like this. And the infected caterpillar that had a modification of behavior, it was this molecular form. You have a glycosylation here on a glycan, something extremely rare in biochemistry. Nobody, well, I don't know another example where you glycosylate a steroid. So how this occurred? Well, it's the virus. The virus acquired by change through evolution an enzyme that allows it to modify, to glycosylate the steroid of the infected caterpillar, thereby hijacking the behavior of its host. It keeps the caterpillar on the leaf of the plant, because if the caterpillar goes down the ground, it's lost for the virus, because the young caterpillar to be infected are on the leaf. So that's evolution and fundamental science. So starting from a guy who had his colony of insect dying in his lab, you end up with a paper in Nature showing that a virus through one single gene can hijack the behavior of a caterpillar. That's fundamental science. Maybe you don't like it. What about application? Well, scientists that were studying the baculoviruses, they were concerned about what we call in virology the host range. The host range is the number of species that a virus can infect. I told you earlier, variola, smallpox, infect human. It has a narrow host range. In contrast, vaccinia has a broad host range. It can infect cow, it can infect human, etc. What about these baculoviruses? Well, they have an extremely narrow host range. There is virtually one baculovirus per insect species. And that's very, very interesting, Why? Well, what about controlling the insects? Pesticides, when you spread pesticide on the field, you have no specificity at all. You kill the good, the bad. You kill the target, the species you want to destroy, but the pure butterfly that comes there and is beautiful is killed like the bad one. And when the bird catch one of them, it get intoxicated. So these are terrible molecules. And so scientists say, so why not replacing these pesticides by highly specific baculoviruses, in which case you will spread the virus on the ecosystem and you will kill like a surgeon just the target species. Sounds crazy? Well, it's done. In the US, you have plenty of biocontrol measure, and you can buy basically baculovirus targeting different species that you can use for different culture. This is science. You start from an observation in an animal facility, a pure guy that is crying because his animal are dying, the subject of research die, and you end up with something that saves the ecosystem. And the beauty is that you don't need to change anything. You take the same plane, you wash the tank, the pipe, and you fill the plane with a suspension of virus rather than a solution of pesticides. And you take off, and that's it. And this works. Now, just to go a step further in the serendipity, I'd like to tell you another example of application of baculoviruses. I mentioned that these baculoviruses, they produce huge amount of protein to unwrap the variant. So scientists thought, well, if they produce a lot of this protein, this virus could produce a lot of another proteins. Why not using these baculovirus to produce antigens to make human vaccines? And the HPV vaccine that some of you have probably used, it's producing like this. So in HPV, this is the variant of a human papillomavirus that causes cervical cancer. Now, this virus has a major constituent, which is called L1 protein. So this protein, if you produce it in large amount and incubate under a proper condition, it will auto-assemble into pentamers and then form what we call virus-like particles. If you give this particle to an electron microscopist, it will tell you, oh, this is HPV. It's not HPV because a lot of components are missing. It's not infection, but it presents the antigen just like the virus. And that's good in terms of vaccination because we can induce a conformational epitope. But these particles that you find in the vaccine of GSK, Cervarix, are produced using baculoviruses, virus from butterfly that have been genetically modified to express this protein. And they are growing, they are grown in insect cells. And nobody knows this. This is a GMO. Nobody gives you this example as a proof that some GMO can be good. They are good and they are bad one. Fourth story. Now the fifth story, which is the last one, and in my point of view, the most beautiful one. So in all the example I gave you, the definition of a good virus depends from which angle you look at. If you ask the algae, are Ficonavirus good? They say no, because they kill the algae. We consider they good because they regulate and save the ecosystem. So it's only on very rare occasion that the host benefit from an infection from the virus. And this is one example. I call this the wildebeest and the alcelaphine herpes virus 1, a rare example of symbiotic relationship between a virus and its host. So for those of you who have knowledge in zoology limited to Walt Disney movie, the wildebeest is the animal that killed Mufasa, the father of Simba. You remember the scene? It's horrible. I don't understand how you can show this to the kids. And this is the scene of the movie Lion King. The wildebeest form huge herd, and it's actually an animal that goes through migration. You walk thousands of kilometers to go where the grass is, and then when the grass disappears, you move away. And this animal, you see these beautiful pictures, and this animal is the host of a herpes virus, alcelaphine herpes virus 1. You know, herpes virus infection Herpes viruses are fascinating virus. They replicate like a normal virus during an acute replication, but then they can establish a kind of dormant infection called latency. So they go into latency, they express very few protein or RNA, dependent of the species, but they are very silent. And from time to time, they can wake up from this dormant infection and induce some replication. Now, the alpha-lacine herpes virus 1 is extremely well adapted to its host. It persists into the host without causing any harm. 100% of the wildebeest are infected. When the cow will deliver the calf, they reactivate the infection, excrete virus that will contaminate the little calf. So the little calf will support an infection, but it doesn't suffer it because the virus has learned through evolution to make a moderate infection. It's adapted with the host. So it looks like the virus is adapted to its host and does not cause harmful effect on its host. So the virus takes benefit from the host to persist and to be maintained through evolution. Now, what could be the advantage for the host to support this infection? Well, here is the beautiful thing, is that while this virus is completely harmful, does not cause any harm to the wildebeest, when the little calf contaminates the other ruminant that are surrounding the wildebeest, this virus causes a lethal disease, an acute disease into the other ruminant called malignant catarrhal fever. And that's very good for the wildebeest because you kill your competitors for grass. And on the top of that, you give prey, easy prey to the lions and to the big carnivores. So you're not only killing the guy that competes with you for the grass, but you are protecting yourself from predation by providing free meal to the lion. And actually, if you talk to Maasai people, they will tell you when the wildebeest are around, you can go in the field without a gun because all the lions are sleeping. They fat like mad. They become fat. They cannot run and they don't have to run because they just receive free meal from the wildebeest provided through this system. And so this is a very, very rare example where you have a kind of a real symbiotic. Symbiotic in biology means the reciprocal beneficial association of two biological entities, where each entity take profit of the association. This is my last story for today. So I don't ask you if you're convinced, but I provide plenty of examples which demonstrate that there are examples of good viruses and they are good by nature, or you can modify them to make a beautiful innovation. Now, I hope I also convince you to be careful when you have to stand up and give an answer in science. It's very, very rare. It's black and white, and you have to be extremely careful. So next week, I will talk about the origin and the evolution and the role of the virus in host evolution. I hope I will see some of you. And before I close, I'd like to very briefly thank again Hans for proposing my name. It's really important for me. I'd like to thank the University of Ghent and Francqui Foundation for this award, and this encouragement come at the right moment. And you know, I'm this year I'm 54, so I've been doing science as a professional for 30 years sharp. You can't believe how fast the time flies. Enjoy every day, because one day you will wake up and you say, what? I lost my hair, I'm 54 years old. What happened? Who moved the clock? And that's very fast. But if I look at these 30 years of professional science, I have to thank all the institution, my university, of course, that I like very much, the FNRS and all these institutions that support me financially. Thanks to them, I've been able to live like the Duve. I have the privilege, and it's a privilege, but also a responsibility to live at this border between the known and the unknown. And every day you wake up with the idea to make the unknown going backwards. And that's a huge privilege. Science also provides another privilege, which is to meet people. It's a teamwork. And as a group leader, you have the chance to see, well, it's a chance, but also sometimes it's not a chance to see people passing in the lab. So So you have the chance to meet a lot of people, and science is really a great human adventure, and I'm very grateful to do this job. And finally, I'd like to thank my family. They're all there. And you remember I talked about my wife. Well, no, I didn't talk about my wife, by the way. Yeah, you remember the blonde one that I mentioned when I was 16? Well, she's my wife. And this shows once more that you can only join the dots by looking backwards. I thank you for your attention.
Okay, Alain, thank you for the lecture. And this was my question: where is the blondie? Which is in the audience. Okay, we don't have time for questions, eh? But I think now it's the let's call it the official things. Don't walk away. You stay here. With great pleasure and also with honor, I can announce you the pro-rector of this university, Professor Alain Vanderplasschen. Alain, the floor is yours. Thank you. Anders gaan we die woorden opzoeken.
Dear Professor Vanderplasschen, dear Alain, Mr. Dean, dear Frank, dear promoter Hans, and then hello to all of you who are here either present or digitally connected. On behalf of the Rector of Ghent University, I would like to thank the Collens Francqui Foundation for giving the opportunity to invite an outstanding, and I think you are all convinced after hearing the lecture, an outstanding international scientist to our alma mater, in recognition of his leading contributions in the world of veterinary immunology and virology. Through his work on different animal species, he acquired an impressive knowledge on the interactions of viruses with their host, which allowed him to develop a broad view on their mutual co-evolution. And I think you will all agree that his lessons are relevant not only to the veterinary world, but to sciences in general. And they illustrate, of course, the important place of veterinary sciences in the medical world. In today's inaugural lecture, and also, of course, the combination of the upcoming lectures that you're going to give later this month, are an opportunity to bring together people from the human and the veterinary medical disciplines. And of course, they provide a unique opportunity for professionals and students to generate interesting discussions among them and exchange their insights on different topics in the field of immunology and virology. Dear Alain, may I say, we feel both very proud and deeply honored that you have been awarded the International Collens Francqui Chair. We would like to thank you for your presence and for your outstanding introductory lecture, which illustrates the beauty of and the fascination for the virological world. You are a very enthusiastic speaker and a scientist who is stimulating the philosophical thinking of young people, I hope. The University of Liège can be very proud to have you in amongst a group of teachers. We are convinced that you already inspired and encouraged many students to observe intriguing and complex phenomena and to come up with plausible hypotheses to explain them. Dear Alain, today I have the privilege of presenting you the Ghent University Medal as a token of our recognition. We sincerely hope that you may continue your outstanding way of doing research, which is exemplary and inspiring for so many young scientists. And finally, I'm very happy to see that your family is also present to share this beautiful moment in your scientific life. I wish you all further success in your professional and in your personal life. And now I'm going to give you the medal. This and it's the connection and the symbiosis of the two, which is important for academics.
Thank you very much.
Yeah.
Thank you. Okay, thank you, Anne. So we don't have the time for questioning. If you want to ask a question, you do it during the reception, because otherwise we won't have a reception. So it's not needed to show the other. So you've seen another 3 lectures coming up. So everybody is mostly welcome to the other lectures. I think it will be very interesting. I knew some of the stories. I knew the story about rabies, because I knew a guy, also a vet, who was in these choppers. He was slipping them around. So that was very nice. So in the name of, not my name, because I'm not going to pay for all of this, I invite you to the reception. The reception is not at the faculty. The reception is outside the faculty, and it's called the Roskamp. If you don't know where it is, follow the crowd. You will be there. Okay. You can go by foot, Hans. Yeah. If you want, if you have problems, you can go by car also. It's not so far away. So first of all, last, I want to thank everybody and just point out the difference between a dean and a non-dean. I have more privileges. I have my necktie and my jacket, but he doesn't have a necktie, but he's not allowed to put off his jacket. So when the reception goes on, you're allowed to put it on. Okay. Thank you very much, and see you at the reception. Thank you.
Don't forget to bring the tickets for the conference. Bring them in front.
If you want to put the ticket somewhere over here,
Then we will select
and
the
then we
winner
will see
later today.
then next week. Okay, great job. Alain.